Okay, hello. Is there any questions?
I have a quick one. In the previous part, you were talking about transcription occurs in the nucleus and translation occurs in cytoplasm, and that's in eukaryotes, right?
Only in eukaryotes, because obviously in prokaryotes, both occur in the cytoplasm because there is no nucleus.
Okay.
Okay? All right, any other questions? Well, if not, let's go ahead and start the screen share. And hopefully more people will be joining, but we'll see. As you can see, there is no lab to do today. We have lab catch up and review. So if you haven't finished lab 4 on stains, please finish that today. And there's a worksheet for that and turn it in. Other than that, I'm going to ask, are there any questions? And then I'm just going to do a little bit of review for the lab. Anyone have any questions? All right, I'm going to go ahead and shut this down. And then go through the review of the lab. No questions at all, huh? Well, in lab 00 on laboratory safety, you should distinguish between safe and unsafe laboratory practices. You don't need to know where the relevant safety equipment is because you're not going into the lab. But you should understand how to safely dispose of contaminated materials and how to dispose of materials. Like if you were in the lab, obviously you're not, but if you were in the lab and you had uncontaminated paper waste, like you were wiping down your table or you're wiping down your scope, you just throw that into the trash can. But if on the other hand, you have bacteria that gets on the paper, then that doesn't go in the regular trash can. You put it in the biohazard bag, which I also call the autoclave bag, because that will be autoclaved before it is put into the regular garbage, okay? And I think that's it for laboratory 00. You should, of course, know how to what to properly wear and what your attire should be if you were to go into the lab. And you should know long hair should be tied back. Absolutely no food or drinking in the lab, and actually no touching of your face with your hands, no chewing on anything like your fingers or something like that. No personal items allowed into the main part of the lab. Obviously, if you bring in things, they go into your cubbyhole, and the region at the door and the cubbyhole are not considered, but considered external to the lab. All right. So that's where all personal items should be. Anything that's brought into the lab proper, it has to be, you cannot take it out of the lab. Okay, it will be confiscated and it will not be returned to you until it's soaked in bleach for 15 minutes or it is autoclaved. Okay, and that would include electronics like your cell phone. And of course, either treatment would destroy your cell phone. Something like paper, especially if it's written on pencil, could be stuck in bleach or autoclaved and then returned to you to take home. So something that is written in ink, that might smear. You should disinfect the lab, your workbench station before you work, and then after you're all done. I already talked about that. So I guess the last thing I want to talk about is when you come into the lab, you should wash your hands. You should put on your lab coat. You should put on gloves. And if you're working with a splash hazard, you would put on goggles. And then you go and do your work. And then when you're ready to leave, if you wore goggles, you take off the goggles and put them in the appropriate place. You take off your gloves, you take off your lab coat, and then you go and wash your hands, and then you can leave the lab. Any questions about that? Well, that's it for reviewing Lab 00. Let's go ahead and review Lab 01. Lab 01 is talking about the ubiquity of microorganisms. Come on. Why didn't that move? So you should know that microorganisms are almost everywhere. You should describe the purpose of culture media, and that's to grow your culture. Describe colony abundance and diversity. I'm surprised that word isn't in there, as well as the morphology of the colony. Be able to describe terms like inoculation, ubiquity of microorganism, what something is, if it's sterile, isolation, incubation, in vitro, that's growing it in the lab, in culture, what a culture media is, and there's 3 types. Well, actually only 2 types. Well, 3 types. We can say a plate and then a tube. The tubes, if it's solid, having auger or some other thing to thicken it, it would be a slant or a deep. And then there's a liquid culture that we call a broth, and they're usually in a tube. Describe what a fastidious organism is, non-selective media. I think we already talked about that, and then what a colony is. All right, any question about any of those terms? Why isn't that working? Ah, okay, I see I have another one here. So the phrase ubiquity of microorganisms means that microorganisms are essentially everywhere. Like you wouldn't find them in a volcanic vent. And if you went into a clean room or a surgery room, you would find that to be fairly clean, but and free of microorganisms. But everywhere else on the planet Earth, generally you would find microorganisms. Something that is sterile means that it's free of living cells, and you would be given media that would be sterilized. And so hopefully the media would be sterile, and it would have no microorganisms in it until you inoculated it. And the purpose of giving you something that's sterile is so that you can grow the microorganisms that you want to grow. So we talked about abundance and diversity of microorganisms, and I'll show you a little picture there about that. You should know a culture medium is something we use to grow our microorganisms in, and there are different types of culture media, and we'll talk about that in another lab. But in this lab, we're talking about there's 2 types of culture medias: a liquid culture media that has that we call a broth, and they're usually in a test tube. And we can add agar or some other solidifying agent to the liquid media to make it a solid medium. And generally, in microbiology, we add agar, but you could also add gelatin or starch to make it a solid medium. Most of the time, it's agar. And then an agar could be in a tube, in which case we call it a deep. If it's just a tube and it's flat across, we call it a deep. If it's a slant, a tube where the agar is in a slant, we call it a slant. The agar gives no nutritional value, it just makes it solid. And generally speaking, bacteria cannot digest the agar. Some bacteria can digest starch or gelatin. All right, any question about any of that? I guess I should say a solid media you can also get in a petri dish, a plate. You usually don't have liquid culture in a plate. And there's a deep, actually better there, and there's a slant. That's probably a liquid broth. You should know that the only media we're talking about really in this lab is general purpose media, and that is a media that is not selective. Its purpose of this media is just to grow up microorganisms. And so it has nutrients to grow up many, but not all microorganisms. And 3 general purpose media that we use frequently are nutrient agar with yeast extract, also called NA, in the abbreviation for nutrient agar with yeast extract. Tryptic soy agar that comes from soybeans, also called TSA, and then nutrient agar, or NA. Okay, so those are 3 general purpose media. They're fairly cheap to use, so we use them a lot. They grow up many microorganisms. They're not selective, but they do not grow all microorganisms. Any question about general purpose media? The purpose is just to grow up things. Everything that you have in the lab should be appropriately labeled with your name, the date, the type of media, and usually you abbreviate that like NA. And then the inoculum, what you're growing in the tube. If you know what it is, you put E. coli. If you don't know what it is, you just say what you did to get it, like air exposure. The incubation temperature is optional. That's usually a good thing to write on there just so that you know what temperature you grew it at. In the lab, we usually grow things at 30 degrees or 37 degrees in the incubator. And when you're writing on a plate, you should always write on the bottom of the plate. You can either write directly on the bottom, and it's kind of hard to describe this, but you can also write on the side of the bottom plate. And the advantage for doing that is you don't have anything that's preventing your seeing the growth on the plate if you write on the side of the bottom plate. But you write on the bottom of the plate, not because that's visible. It's true, we do grow things upside down in the incubator, but you write on the bottom of the plate because that's where the information is, and the bottom of the plate is where your culture will be. And if the lid gets separated from the bottom of the plate, you want the information with the bottom of the plate. Any question about any of that? Why we grow things upside down in an incubator is that, at least the plates, they have media in them that contain moisture, and in the incubator, the moisture, if you were to grow it right side up, the moisture would come out of the agar and then condense on the lid of the plate and then drop back into the culture. And that would spread what you're growing in the culture, potentially ruining the culture. And so we grow them upside down. And that way, if the moisture comes out of the agar, it will solidify on the top, which is the agar, and that will wick the moisture throughout the agar, so it will not drop down. And then if it did drop down, it wouldn't be a problem because you don't have any culture growing on the lid. Any question about any of that? All right. right, I think that's it, except for talking about colony abundance and diversity. I guess you should, well, no, I'm not going to ask you to tell me, is the elevation of the plate flat, raised, convex, or umbunate? I wouldn't do that. So I'm not going to ask you to memorize that. But you should know the abundance and diversity. Like the abundance in this plate is, or excuse me, this picture is 1, 2, 3, 4, 5, 6, 7. There's 7 animals shown, 5 cheetahs and 2 lions. The diversity is 2. There's 2 different cats, lions and cheetahs. All right? And you could describe the colony abundance and diversity of this plate here. And you were asked to do that in the worksheet. A number of you kind of got that incorrectly. How you determine the abundance is you just count these, and I would say there's about 30 colonies there. I'm not going to do the actual count. For the diversity, you look on the plate, and if the colony looks different, you count it as one species. Like those 2 colonies actually look slightly different. So that would be 2 species. That colony is smaller than those 2 and also wider. So that would be 3. This one's much bigger, and it also looks different than that colony and those colonies. So that's 4. This one's a different color, so that's 5. This one's a really different shape, although that could be the same as that. So we'll just say 5. This one's a different color and much bigger than all of these. So that's 6. And then there's one that's much bigger. And so there's at least 7. Actually, there's another one that's different. So that would be probably at least 7, probably at least 8 for the diversity. And the abundance, something like 30. Okay? Any questions about any of that? All right, that's it for lab 1. Why is that taking so long to shut? Let's move on to lab 2. Come on. Looks like I should open these. You should know what the purpose of aseptic transfer is, and that you should use aseptic technique when you're working in the microbiology lab. You should use that also if you go and work in a healthcare facility. Determine if aseptic transfer technique was used from the data provided, and define the terms ubiquitous, aseptic technique, inoculation, contaminant, and something that's sterile. Oh, good, I'm puking there. You should know that pure cultures were something that Louis Pasteur came up with, and a pure culture is something that contains only one species of microorganism. And when you're working in the lab, of a microbiology lab, you generally should be working with pure cultures, because if you have a mixed culture, one with more than one species in it, and you get a positive result, you don't know which species gave you the positive result. So you should be working with a pure culture. And Louis Pasteur came up with the concept of the pure culture and then how to obtain a pure culture. He did that by limiting dilution, meaning he diluted out until he got something growing in only 1 of 10 tubes of a limiting dilution. And it was a very difficult way to get pure cultures. So we no longer use that way to make a pure culture. But Louis Pasteur came up with the concept of a pure culture. And then he came up with aseptic transfer. Louis Pasteur knew that the air contained microorganisms. And if you wanted to grow a pure culture, you had to use aseptic technique in order to keep your culture pure. And you do that by keeping it from being contaminated from the accidental inoculation of unwanted organisms, either from the environment or from the air. And so when you're growing up your puriculture, you put it into a sterile media and you use aseptic technique where you sterilize the instrument you're going to use to transfer the culture, the organisms, into the new sterile media. And then you cap that sterile media as soon as you're done inoculating it so it remains uncontaminated. Any question about any of that? And so in the lab, you worked and you did that. You did it virtually. You sterilize things. In our lab, we would use a Bacto incinerator to sterilize a loop. Is that shown above? You can sometimes use a flame to sterilize your loop. There's the loop there. And you can only sterilize the wire portion of the loop. You cannot sterilize the handle portion of the loop. Yes, somebody had a question. No? All right. So you sterilize the wire portion by putting it either in the flame or the Bacto incinerator. When it turns red, you know it's sterile, and then you bring it out. And you have to let it cool before you pick up your microorganism.
What was the answer to the question when it said that it sizzled when it was placed into the medium? One of the lab questions.
Yes.
Why did it? Is it because the loop was too hot?
Yeah, the loop was too hot. You're supposed to cool it before you pick up microorganisms. Okay?
So that denatured it, right?
That initially killed all the organisms that he picked up on the loop, but
oh, wow.
Probably noticed that, and then he picked up more organisms on the loop, and only the first time would it kill the microorganisms, because that would cool the loop. And usually what you want to do is cool the loop before you pick up the microorganisms. And how you do that, you could simply hold the loop in the air for 20 or 30 seconds and just let it air cool. That's not the best way to cool it, because during that 20 to 30 seconds, something from the air could touch the loop, but that does work. And in 20 seconds in particular, you're unlikely to get contamination from the air. The other way you can cool the loop is you open up the tube that has your microorganisms in it, and then you touch the loop 4 times on the side of the loop, and that will cool the loop. And then you pick up your microorganisms and transfer them into your sterile media. That's the best way to cool your loop, because it happens immediately. It should be sterile inside your tube. And then you pick up your microorganisms and you carry it through your sterile media. Okay, I don't think there's much I need to talk about here. Let me remind you that when you're reading the meniscus, the liquid in either a pipette or a tube or a burette, it will be slanted because the liquid attaches better to the glass than it does elsewhere. And you read the meniscus at the bottom of the meniscus, not at the top, not halfway in between. Generally, when you inoculate a slant, you do it in an S-type motion. And so you start at the bottom of the slant or the top, it doesn't matter. And then you move up the slant in an S-type motion. And some people didn't seem to get this right. The questions P1, this is P1, this is P2, that's P3, that's P4. This is turbid. This is a clear slant or clear liquid broth. This is a turbid liquid broth that's pink because Serratia marsens was growing in it and it's kind of pinkish. And then this is a slant, and you can see red on the slant. That's Serratia marsens. It's a reddish color when it grows on the slant. And anyways, that was that. All right, any questions about lab 2? If not, let's move on to lab 3. On the microscopes. You should, of course, with the microscopes, understand the different parts of the microscope. You have to know all the parts of the microscope, but the parts we use, and it's on the figure. Calculate the total magnification for an objective lens. You should understand how to focus the specimen using the different objectives. And you always start with the scanning lens. Start your focusing there, and then move the specimen to the center of the field of view, and then move up to the next lens. That would be the low power or the 10x lens. Get it in fine focus, and at this point you should only use fine focus, and then move the specimen to the center of your field of view, and then move up to the next lens, which is called high power, but that's the 40x lens. Use your fine focus only, move it to the center of your field of view, and then move that lens halfway out of the way. Don't move the oil immersion lens in yet. Add your drop of oil, and then move the oil immersion lens into that drop of oil. You should only use fine focus and finally focus on your specimen. Okay? One of the questions was to estimate the size of the specimen, and you can do that by knowing the size of your field of view. Depending on which lens you're doing, I'll show you the table of that. Understand the proper use for proper microscope care. We expect you to know that, but obviously you're not going to be using it because you're not actually going to be using microscopes. But microscopes are expensive and they have to be used appropriately and then cleaned appropriately after you're done with it and then put away. So here are the parts of the microscope you should know, and they're labeled here. You should know the different magnification lenses, what their magnification is for the objective lenses. And for our scopes, you would take this lens and then multiply by the ocular lens to get the total magnification. And all of our scopes have a 10x ocular lens. And so the total magnification would be this for our objective lenses. I won't hold you to know the names of the different lens. Like I won't ask you a question like, which objective lens is the low power lens? Okay, so you only need to know the magnification power for the objective lenses, and then the total magnification. You should know the depth of field, that with the 4x objective lens, it has the deepest, biggest depth of field. And then as you go up in power, the depth of field becomes smaller. And so the smallest depth of field will be the 100x objective lens. And that same thing with the field of view. The field of view, what you're seeing under the microscope, will be largest with the 4x objective lens. And as you increase the power of the objective lens, you'll have a smaller and smaller field of view. And that means the 4x objective lens is the easiest lens to work with. And then the 100x objective lens is the hardest to use. Any question there? So you should know how to use a microscope, how to get something in focus. I thought about this, and I don't think I'm going to require you to memorize the diameter for the objective lenses. I will provide it to you if I want you to be able to use it, okay? But from this, you should be able to estimate the size of a cell under the microscope. For example, if a cell like, I don't know, a frog egg, would that be about that? No, I can't think of an example, but if the specimen fills up the whole field of view, I'll give you this table. When we're looking with the 4X lens, then it would be close to 4.5 millimeters in diameter. Any question about that? So you don't need to memorize that. I'll provide it, but you should know how to use it. We do expect you to know how to use a microscope and then view prepared slides. You can only do this virtually, but we do expect you to know how to do that. We're not going to ask you anything about making a wet mount, but you should know how to put a microscope away, lower the stage completely, remove the slide. If you used oil, the oil has to be removed from the lenses, and you should check both the 100x lens if you use oil and the 40x lens, because if you're rotating the 40x lens around, if it touches the oil, it will pick up oil. And usually when students use oil and then they switch around, they just move the objective lens and they might not notice that the 40x lens came into contact with the oil. So you should test both lenses for oil and use only lens paper to remove oil from the lenses. Lens paper, let me mention a little bit about that. It's paper that has had the wood fibers removed from it, and so it will not scratch the lens. Chem wipe, tissue paper, paper towels, any other paper has wood fibers in it, and it will scratch the lenses. So only lens paper should be used on the lenses. And then put the 4X lens in place when you're done with the microscope. Lower the light intensity, switch off the light, unplug the microscope, and wrap the cord around the microscope, and then put the microscope away the way it was taken out. And generally, the cord is facing you on the microscope. I'm going to have you do number 7, so we won't talk about it. Oh, when you're carrying the microscope, you should generally hold it with 2 hands to keep it safe. And I already said that. Put it back the way you got it. All right, any questions about this lab, lab 3? If there's no questions, we'll move on to our last lab, lab 4. And this is staining specimens, working with stains. So you should know how to prepare a bacterial smear before you're going to stain it. And generally, a bacterial smear should be very thin. Ideally, you want the cells put down on the slide in only a single layer. Understand the principles and procedures for performing a simple stain, a differential stain, and a special stain. interpret the results from a simple stain, a differential stain, and a special stain, and describe the following terms. Okay? Any question about any of them? We use a stain for our specimens because if we did not, they would look very close to the glass of the slide, and it would be very hard to distinguish the bacterial cell from the background of the slide. And what we do is we use a stain to increase the contrast between the cell and the background of the cell. And it doesn't matter if it's an acid stain or a basic stain, it increases the contrast. The difference is a basic stain stains the cell and not the background, and an acid stain makes a negative stain, and it stains the background and not the cell, but it still increases the contrast, and so that way you can see the cells better. A simple stain is just a stain where you use one dye to stain the cells. Any question about that? So you can't distinguish cells except by their morphology. Like you might be able to distinguish a diplococci from a staphylococci, but everything would be the same color. You could also distinguish a bacilli from a cocci. But once again, they'd be the same color in a simple stain. A differential stain allows you to differentiate between cells, and it uses two or more dyes to stain the cells. Differential stains, the best one that we use is the gram stain, and it differentiates cells based upon the makeup of their cell wall. There are different differential stains, but like I said, probably the only one you need to know about is the gram stain. And lastly, there's a third type of a stain, the first being a simple stain, the second a differential stain, and the third type is a special stain, and it allows you to see or stain special structures in the cell. Like you could have a capsule stain, or an endospore stain, and a Flagella stain. Actually, these two aren't good examples of a special stain because the Flagella stain is the only one that's specifically a special stain. An endospore stain, you could use a special stain to see endospores, or you can also use a differential stain to see endospores. Is that clear? And the same thing with the capsule. You could use a special stain to see the capsule, and that generally is only using one dye like India ink. But you could use a differential stain to see the capsule. And so like I said, these aren't good examples for a special stain because there are differential stains to see a capsule and an endospore. Any question about that? I think in this lab, we're using a special stain to view endospores and capsules. The gram stain was invented by Christian Graham, and so when you spell it out, you should use a capital G because it's named after somebody. It uses crystal violet as the primary dye, and it initially stains all cells purple. Okay? It separates cells based on their cell wall, and the gram-positive cells will retain that primary dye, the crystal violet, and stay purple. The gram-negative cells lose the primary dye, the crystal violet, and they lose the purple color, and that's why they will stain pink in the gram stain, and they're pink because the second stain used, the counter stain, safranin, is pinkish in color. The gram stain stains the cells based upon their cell wall. The gram-positive cells have a thick peptidoglycan layer, and the crystal violet and the iodine gets stuck in this, and it doesn't wash out in the decolorizing step. In the gram-negative cells, they do have peptidoglycan, but it's thinner, and it's thin enough that the crystal violet iodine complex washes out of it. And that's the basis of the staining for the gram stain. In the gram stain, we do use Graham's iodine, which is a stain or a dye, but we're not using it that way in the gram stain. We're using it as a mordant. The iodine combines with the crystal violet to make a larger complex. And so really it's the crystal violet iodine complex that gets stuck in the gram positive cell wall and does not wash out. We then use the decolorizing agent, which is usually alcohol acetone combination, and it will pull out the crystal violet iodine complex from the cell wall of a gram-negative cell, but if done correctly, will not pull out the crystal violet iodine complex of the gram-positive cell. And then we use the counter stain, safranin, and that will dye the gram-negative cells pinkish in color. Okay, any question about any of that? I don't have a slide showing you the different steps of the gram stain, but you should know after the decolorizing step that the gram-positive cells are still staining purple. The gram-negative cells at this point are clear, and they do not become pink until you add the safranin dye. Any questions about that? Now, the gram stain essentially separates most bacteria into gram-positive and gram-negative cells, but there are some bacteria that occasionally don't stain well, if at all, by the gram stain, and that's the Mycobacterium. This family of bacteria have a lot of lipid, it's mycolic acid in the cell wall, and the mycolic acid prevents the dye from getting in the cells. And so they may not stain, or they stain lightly. They're actually gram-positive, so they're supposed to stain purple. But sometimes when you stain them, they will appear gram-variable, where some cells will stain purple and other cells will stain pinkish, okay? It's really only two families, the Mycobacterium and Nocardia, that we're not going to talk about, that don't stain well with the gram stain. All right, but if they have a cell wall, they'll stain gram-positive or gram-negative with the gram stain. The exception being the family really Mycobacterium. Any question about that? The gram stain, there are problems of doing the gram stain. First of all, you should only use freshly grown cells. If the cells are older than 2 days old, you shouldn't use the gram stain, or you shouldn't stain them with the gram stain, because the stain may not happen appropriately. Every time I've found students who had a result like this, where the cells were staining two different colors, what we call gram-variable, it wasn't because they were staining Mycobacterium. It was because they were staining old cells, at least a week old, some cases 2 weeks old. And what's happening there is that the cells that are viable are staining gram-positive. The cells that are dying or non-viable are staining gram-negative. And the gram stain does not work well if the cells are old. You should prepare a thin smear, because if it isn't thin, you'll have trouble getting the dye all the way through the layers of cell. And then in the decolorizing step, you'll also have trouble removing all the dye if it should be removed from all the cells in many layers. If you heat fix the bacteria too severe, meaning too much, you will not have trouble with the coloring, but you'll have trouble with the shape of the cells. And then bacilli, if they're overheat fixed, they won't appear like bacilli. The cells will melt, and if they melt, they'll shrink in their shape and become cocobacilli. I'm not sure what cocci would look like if you melt them. I'm just not sure. You should use fresh staining reagents, and then the most tricky step is the decolorizing step. If you over decolorize, you will pull out crystal violet iodine from the gram-positive cells, and if you under decolorize, you will not pull out crystal violet iodine from the gram-negative cells. Any question about that? All right. There's an acid-fast stain. That's actually another differential stain. I'm not going to talk about it. I won't quiz you on acid-fast stains, but you use that to find mycobacteria, which stain pinkish in the acid-fast stain. Special stains, you can use a special stain to look at the capsule, like if you use India ink. I don't think this is India ink, but it says it is. The reason why it doesn't look like India ink to me is it is staining the background, but it's staining the cells much more intensely than the background, and the capsules are not being stained. So in that sense, it does show you a capsule like India ink it would. But India ink stains the background darkly, and then the cells are really, really lightly stained. That's why I really don't think that's India ink here. I think this is stained with two dyes. The first dye stained the cell, and then they're using India ink to stain the background. Okay, any question about that? There's also staining for endospores. Let me state that endospores are mostly made by two families of bacteria, Bacillus and Clostridium. An endospore is something the parent cell makes when the environment is becoming detrimental to the survival of the parent cell. And then when the environment becomes so detrimental that the parent cell dies, if the parent cell made an endospore, the endospore can continue living, because endospores are very hardy. They're the hardiest thing we have as far as life goes, and they can survive most environments, okay? So they're very hardy and survive very well. Any question about any of that? You do not have to memorize this table. And generally, when we stain for endospores, let me see if I can blow that up. The endospore generally stains greenish, and this is an endospore outside of a cell. There's an endospore inside a cell, meaning the cell is forming it. It hasn't broken away from the cell yet. The cell isn't dead. And you can see a little bit of the cell on this side. Most of the cell is on that side of the membrane. On that one, it's better. Let's see if I can blow that up just a little better. You can see an endospore in this cell, and the parent cell is on both sides of the endospore. On that cell, the endospore is near the end of the cell, and so there's very little cell at the end. It's mostly an endospore at the end. Any question about any of that? There's a special stain to show the flagella, shown here, and that's called the Flagella Stain. shown here. Okay, know the terms for laboratory exercise 4. From a simple stain, all the cells will be the same color, but you might be able to distinguish differences in the morphology, the shape of the bacteria, or their cellular growth arrangement if it was grown in liquid culture. You cannot distinguish between the color of the bacteria on a simple stain because they're all going to be the same color. On the gram stain, oh, here we go. When you're going through the gram stain, when you add the crystal violet, all the stain cells will be purple. When you add the grams iodine, all the cells will be purple. They're generally a little bit more intense purple or a darker shade of purple. When you add the decolorizing step, add the decolorizer, the gram-negative cells will stain clear. Gram-positive cells are still purple. And then when you add the safranin, the gram-negative cells will be pink, and then the gram-positive cells will be purple. And you can distinguish between the different cells by their color in the gram stain. All right, any questions about this lab?
Did you go over the last two or not yet? Because maybe not everybody's submitted in yet.
Want me to go over the last two stains?
No, the case study.
Case, case study 1. All right, this is a 45-year-old female experiencing a respiratory issue based on the clinical presentation. In the worksheet, hopefully I've got a picture of a normal lung. Anyways, it's showing you her clinical presentation. The doctor orders a chest x-ray and sputum from the patient, and then they use AFB, which stands for acid-fast bacterial stain. Okay, and let's go to the worksheet. Hopefully you saw that. Yeah, I got the normal lung picture in the worksheet. The normal lung is clear and the lungs look dark. And here you see we've got a splotch right here and right there that is not dark. And so something is probably in the lungs creating the undark spots on the x-ray of this individual's lungs. Any question about that? And then when we look at the acid-fast stain, it tells you they followed the Ziehl-Neersen stain. We're seeing red cells. So these are acid-fast positive. And then there's also dark cells, and that could be a dark cocci there. These are probably cells from the patient, meaning they're their cells, eukaryotic cells. And they're not acid-fast positive, they're acid-fast negative. And if we see acid-fast positive cells and an acid-fast stain, it means you have mycobacterium. And so this patient has an infection in their lungs of Mycobacterium tuberculosis. Now we haven't specifically identified that it's Mycobacterium tuberculosis. We haven't done the test to determine that, but it's in the lungs and it's acid-fast. And so it's Mycobacterium in the lungs. And so it's most likely Mycobacterium tuberculosis because that grows in the lungs. Any question about that? That's case study 1. Case study 2, 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis, shown here. A routine culture stain and a gram stain is ordered because the bacteria grew anaerobically. I'm not sure how they know that it's growing anaerobically, but we'll take it that way. Based on the findings below, what's the most probable diagnosis? Well, we see in the gram stain that they're gram positive. We have bad tissue necrosis here, and it's given that the bacteria are growing anaerobically. And then when we do the Schaefer-Fulton stain, the stains for endospores, we notice green staining endospores from the pinkish parietal or vegetative cells. Any question about any of that? So the bacteria is gram positive, it's creating endospores, and it's growing anaerobically, and it is causing tissue necrosis. So if you come up to this table here, oops, I'm in the wrong one, just a minute. Where do I have that? If you come up to this table in the lab, it's one of the species creating endospores, so that means it's most likely bacilli or a clostridium. Because bacillus grows aerobically, then we know it's clostridium. And you can't tell from the endospores which one it is, but you look at the 4 clostridium, this is causing food poisoning. That's not what the patient has. It's not causing tetanus, so that's not what the patient has. Gangrene, that is a possibility. Gangrene causes tissue necrosis, and if you look at pictures of gangrene online, it would look similar to the foot of that patient. And so it's probably Clostridium perfringens. And it's not colitis, that would be gastrointestinal distress. So it's not Clostridium difficile. All right? Any questions
Thank you.
about that? All right? All right, that's it for the lab. Are there any other questions? If not, I will see you next time. Bye.